all our respected seniors who are joining us today. Welcome to today's meeting. Welcome to today's virtual CME. Our CME today is on regenerative. Our CME today is on regenerative stem cells. Just 1 minute. Perfect. Perfect. Karibuni sana, esteemed members of the Pharmaceutical Society of Kenya, respected seniors who are joining us today. Today, our webinar is on regenerative medicine and stem cells. Our speaker for today shall be none other than Dr. Juliet Makanga, and our meeting shall begin in a short while. So probably, if I may ask, where are you joining us from? I want to know where you're joining, which part of the country you're joining us from, and which branch of PSK. Don't write part of country. Probably on the chat box, write the part of the branch of PSK that you belong to. This is so as to buy time. I want us to probably get to 250 to 300 attendees. We're going to do so that we're able to kickstart our meeting for today. Kindly on the chat box, let me know which part of the country you are joining us from. The branch of PSK that you're able to join us from. We begin in a short while. Once again, wonderful good evening to all our esteemed members of the profession, seniors of the Pharmaceutical Society of Kenya. Welcome to this meeting today. Our meeting today is on regenerative medicine and stem cells. Dr. Juliet Makanga will be taking us through. Probably, it seems a lot of members are either stuck in traffic or so, because I can see we are yet to get to 200. I can see right now the members who are joining us are from Western Branch. Welcome very much, Dr. Joan Kadesa. Well, karibu sana, senior respected member, Dr. Alfred Birishi. Perfectly well to see our seniors in here today. Dr. Ashok Shah, karibu sana from Kisumu. Dr. Hakim Kiboya, young pharmacist, karibu sana from Nairobi Branch. Dr. Shauri from Coast Branch, Dr. Stephen Oshira, Central Branch, karibuni sana. Dr. Jitesh, karibu sana from Meru Branch. We're much delighted to host you, and it's an honor. Karibuni sana, ladies and gentlemen. Dr. Kwansan Demo, karibu sana, Daktari. Hope you join Nairobi Branch soonest possible. Perfect. Dr. Kibwongo Gitahi, karibu sana, respected senior member of the Pharmaceutical Society of Kenya, joining us from Nairobi Branch. So, esteemed members, it seems today members are joining in slow. I'll need to activate and remind them to join us for today. So we begin our meeting for today. Just before we begin, allow me to take us to take us through some few ground rules. First and foremost is to thank you for participating in this session. All participants are muted during the entire course of this session. Please ask questions via the Q&A tab at the bottom of your window. Questions shall be addressed by presenters on the Q&A tab. For PSK members, if not already done so, kindly subscribe onto the on the PPB portal for this meeting. It is on the PPB portal. I hope majority of you have been able to see it. Right now, the portal has been reactivated. The portal is quite fast, and today's meeting, as it appears on the portal, I want to get you the name as it appears on the portal. It appears on the first page, on page 100, and it appears as Regenerative Medicine and Stem Cells, a new era in drug discovery and development of pharmaceutical products. For KPA members, the attendance list shall be shared with the officials for awarding of CPD points. A point to note is that this session is being recorded and will be made available on PSK's YouTube page. And probably just a quick ask, how many members did the survey for the conference, and are you willing, do you know the dates for the conference, for the upcoming Pharmaceutical Society of Kenya annual conference? Do you know the dates? So this is the agenda for today. Our agenda for today is welcome and opening remarks being done by myself, the presenter, by none other than Dr. Juliet. Then moderation is going to be done by my colleague, Dr. Eric Muringu. Also, close of the meeting by Dr. Eric Muringu. So, ladies and gentlemen, let me stop sharing screen and allow Dr. Juliet share her screen. Dr. Ari, if you're able to share your screen, I'm waiting for you to share your screen. Perfect. So, ladies and gentlemen, without further much ado, let me now take this opportunity to welcome none other than Dr. Juliet Makanga. She is going to take us through regenerative medicine and stem cells. She is a lecturer at Kenyatta University. I will not speak much about her because I will probably be preempting her presentation. She is a wonderful lady who inspires both the young and old. Karibu sana daktari.
Thank you. Good evening, esteemed members and senior respected members. As been introduced, thank you for the introduction, Daktari. I plan on discussing a little bit about regenerative medicine and stem cells and how it impacts us as a pharmacists and even in the pharmaceutical industry and development of new products. I hope I'm loud and clear.
Okay. Yes, you are loud and clear, Daktari. You can go ahead.
Okay. Just hold on. Give me a second. Sorry. Just please hold on a second, members. I'm having a little bit of a challenge here. Okay, I hope now it's clear. So let's get a little bit of the terms that will be the main concept or we'll be addressing as we go by. First of all, regenerative medicine. Now, regenerative medicine is described or defined as a branch of medicine that involves regrowing, repairing, or replacing damaged or diseased cells, organs, or tissues. So here you can imagine a patient who's had a stroke and lost function in some part of their brain, whereby they are paralyzed, for example, and we are hoping to replace the damaged cells with other cells that we have generated in a laboratory somewhere. So regenerative medicine includes not only the generation and the use of therapeutic stem cells, there's also tissue engineering, whereby if you think about it, maybe 20 years from now, we will no longer need to have donor waiting lists. We'll be able to generate tissues or engineer tissues in the lab. and also production of artificial organs. So in summary, there we have the tissue, excuse me, tissue engineering, biomaterials, of which now we as pharmacists will clearly fall into this docket, and also stem cell therapy. So I hope by the session we will understand stem cell therapy more. Okay, for some reason, I'm sorry about that. Great. So what are stem cells? Now, I know we all know this, but just as a reminder, stem cells are undifferentiated cells, and they have the capacity to multiply. So they have the replication capacity and capacity to differentiate to various cells of the human body. So we can rephrase this and say stem cells are pluripotent. Okay. So when it comes to regenerative medicine, it actually encompasses quite a number of other elements. I know we all are conversant with personalized medicine or precision medicine, whereby we now come up with drugs or medication. We tailor-make medication for specific patients. It could be because of the genotype of the patient or the genotype of the disease they're having or whatever it is. So now this can include, of course, stem cells or other drugs, especially, for example, for the breast cancer, whether it is estrogen receptor positive or not, such as the precision medicine, that is still under regenerative medicine. Then we do have stem cell therapy. This is where we are using stem cells to treat disorders or certain conditions, very commonly used in spinal cord injuries. We are talking about things such as Parkinson's disease, and we'll see a little bit about that later on. And lastly, this is now the stem cell drugs, which is a more updated and upcoming part of regenerative medicine, is where we are creating, commercializing the stem cells or some stem cells products, and being able to create commercial off-the-shelf products with industrial production. Of course, in this case, we'll have to follow GMP procedures and the rest. So this is, I think, where we come in. Of course, as pharmacists, we will play a role all through, but now there's the new concept of stem cell drugs and not only stem cell therapy. So a little bit of a history about stem cells. Now, we are in 2001 right now, and the whole idea of stem cells isn't very old, considering that the first time the existence of cells that multiply, replicate, and differentiate in the body, which was demonstrated by the existence of hematopoietic cells, was in around 1978. So it's not very old, the whole idea of stem cells. This was followed by isolation of embryonic stem cells. And we're going to see why this term here, embryonic, is very crucial. And the first ever isolated embryonic stem cells were from the mouse embryos, the rodents, okay? Then we followed through a path and ended up with, and we'll discuss Dolly, the clone. Okay, Dolly, the sheep that was cloned, was another very interesting discovery when it comes to stem cells. But only in 1998 did we have the first human embryonic stem cells being harvested and cultured. After that, in 2008, we had the first clinical trial that was based on human embryonic stem cells, and that was addressing spinal cord injury patients. Then lastly, finally on this slide, we do have something mentioned about another set of cells that are going to be extremely interesting, as we are going to see. Now, these are called the iPS cells, which come in to kind of help us out with some of the disadvantages. And I'm sure already when we mention embryonic, most of us are thinking about the ethical issues that could be associated with the kind of therapies that I am trying to talk about today. So all in all, this is a relatively new aspect of medication or treatment, sorry. So cloning and Dolly. What was so special about Dolly? What Sir, and he was knighted by, of course, the Queen, and hence it's Wilmot, and his team were able to do is to take a cell, a donor nucleus, and now from this is a sheep's udder. A sheep's udder is a mature cell, a somatic cell. They took the nucleus of a somatic cell, fused it with an egg that did not have any nucleus. After the fusion, they cultured it, okay? They fused and formed an embryo, which was implanted into a foster mother, and we ended up with a cloned lamb, Dolly. So Dolly is ideally a clone of the donor cell sheep. What was interesting here is it was as if we were able to reverse the process of differentiation. I will give another example. Now, before Dolly, and actually many of us believe that Dolly is the only clone animal available. Before Dolly, there were actually 9 other clones. What was unique about Dolly is they did not make use of any embryonic cells. Okay, they only made use of somatic cells. So it was very interesting that within the nucleus of a somatic cell, there seems to be information that is able to create a whole organism. Okay. Now this brings me back to, or rather takes me back a couple, not really a couple, but quite a number of years before. Even before Dolly was created, we have Sir Gordon, another one. He was knighted and ended up receiving the Nobel Prize in Physiology or Medicine in 2012 for this study. What Sir Gordon did is he took an intestinal cell and he was using a frog, irradiated an egg, which means now that it didn't have the nucleus, introduced the nucleus of the intestinal cell into the denucleated egg. And he ended up creating a cloned frog. So what we're saying here is by replacing the nucleus with the nucleus in the egg taken from a somatic cell, he was able to grow a new frog. So what this kind of created the idea that a mature cell, a somatic cell, still contains genetic information that is needed to form all the types of cells to form any organism. Okay, so this was actually prior to Dolly. So the first cloned animal actually isn't Dolly, but is the frog that Sir John Gordon created in 1962. But now, why the hullabaloo about these stem cells? We've managed to make them with Dolly animals, and there have been a myriad number of cloned animals, from pigs to, we have the bovine, we have the porcine, but we still had a big challenge with the human embryonic cells. But why are we targeting the human embryonic cells? Why do we need to culture them? Why do we need to isolate them? Okay. So from the basic biology of the embryonic stem cells, as I had mentioned in the start, they're able to replicate, self-replicate, self-renewal, and also differentiate to all the cell types that make up the body. So if we are able to do this in vitro within the dish, in vitro, they are able to differentiate. We can observe the processes of differentiation. So we are able to study developmental study and how the genes affect early development. Apart from that, if we create these cells, mimic them in the dish, we are able to study drug efficacy and toxicity. And then if you think big, we're able to create organs that can be used for transplantation. So this is actually what accelerated the study of, or the study into embryonic human, that is human embryonic stem cells. Because for other species, they have always been easy to isolate and culture, but for the human embryonic stem cells, that has been evasive or was evasive. So to take us back to our first year of, I think this we did during anatomy. We do have fertilization, of course. There again, the sperm, we have formation of the zygote, and then we have the formation of the blastocyst, okay, which contains the inner cell mass, outer cell mass, the cavity. Later on, we will have the ectoderm, mesoderm, endoderm, and of course, different parts of or different tissues and organs differentiating from the 3 germ layers. And what I'm going to focus on is on this blastocyst. So ideally, embryonic stem cells are isolated from, in the case of a mouse, it will be embryonic day 3. For the human, it's embryonic day 5. Okay. And that is from the inner cell mass. Okay. Here we have the embryonic cells. So within the body, what would happen is the embryonic cells are the only ones that possess what we call totipotency. Totipotency is slightly different from pluripotency, because pluripotency means that a cell can differentiate to almost all, but not all, cells of an organism. The cells that are normally exempted when we are talking about human ES cells or embryonic stem cells are the germ cells, the sperm and the egg. So the only cells in the human body that are able to actually produce all cells, including the gametes, are the ES cells or the embryonic stem cells. I hope I'm clear till there. So if we could isolate or get the human embryonic stem cells and create cell lines, this should be useful to study the human development, of course, biology process. Of course, I've mentioned drug discovery and also ultimately transplantation medicine. So we are moving the embryonic stem cells from the in vivo to the in vitro. No, sorry, from the in vitro to the vivo in the plates. Now, this was successfully done by Thompson and his team, and this was very recently. I think should still have been in primary school by then, in 1998. This was the first time human embryonic cells were isolated, and not only isolated, but successfully cultured and cloned, which means they could be stored and used for experiments in a laboratory. So here is some images from Thompson and his work. So this is the inner cell mass that was, they derived the cells from those. And normally the embryonic stem cells or stem cells in general are grown on top of a layer of fibroblasts. They're really, in biotechnology, we call them very needy cells per se. And okay, from what they had isolated, they were able to clone different cell lines. And these different cell lines, when exposed to different conditions, were able to differentiate. You can see here a change in morphology of the cells. Now you'd wonder, what was the source of these human embryonic stem cells? So for Thompson and his team, in this case, these were IVF, pre-implantation embryos that were obtained with the informed consent and for the purpose of this experiment. Okay, so that already should be creating a red flag in our heads for some sort. And that brings me to the next slide. The dilemma that we had here now is it morally acceptable to pursue all these novel therapies that we want to study the process, and maybe cure illnesses, yes, but at the expense of destroying early human embryos? So are we creating embryos only for the purpose of studying disease? This has been a bone of contention, of course, ethically and also politically, in the sense that leading to a lot of change in legislation in different countries, where the fertilization of a human or production of human ES cells simply for the purpose of experiments has been banned. So there's the ethical aspect, of course, and it is quite complicated. So this created a problem. What next? We know human ES cells are useful, but are we going to stop researching them because of the ethical and the moral aspect of it. So they came up, the next step was to think outside the box. We have human embryonic stem cells, very useful, but ethically questionable. Now, this brings us to the development of what we are calling induced pluripotent stem cells. Here, of key importance to note is the term pluripotent. We are saying they are not totipotent, they're pluripotent, so they're stem cell-like. Okay, but they're not 100% same to or similar to the human ES cells, as we are going to see. So iPS cells. Induced pluripotent stem cells, these were generated or established by Yamanaka, pictured here, who also received the Nobel Prize alongside Sir Gordon in 2000 and, what did I say, 2012, was that? Yes, so in 2006, Professor Yamanaka sat and thought, what if I forcefully reminded a somatic cell that it used to be a stem cell? And the way he did that was to forcefully express certain genes, genes that are normally specifically found, turned on or expressed in stem cells, and that is embryonic stem cells. So his studies first started with the rodents, he started with mice, and he was able to establish cells that were very similar to the embryonic stem cells in character by introducing only 4 factors. These factors are now commonly known as Yamanaka factors. Okay, I'll explain a little bit more about them. So iPS cells here, or the induced pluripotent cells, are now considered morally superior to the embryonic stem cells because now we are not using embryos to generate them. All we have to do is to have a somatic cell, whatever cell it is. And we're going to see we use skin cells because they're easy to isolate, just a skin biopsy, and do not require any destruction of embryos. So what Yamanaka's work actually did, and why they actually shared the Nobel Prize that year, is he was able to prove what Sir Gordon had said initially in his work in the 60s, that a mature cell still contains genetic information that is needed to form all cell types. What we're saying is a somatic cell still has stemness. All you have to do is to switch on the stemness. And that is what Yamanaka did with his 4 transcription factors. So this, I'm going to show you some of the work I have done. So Yamanaka first produced his cells from mice. We worked on rats and were able to isolate rat fibroblasts. These are rat skin cells, fibroblasts, and introduce the Yamanaka factors using electroporation. Okay, there are 4 factors, transcription factors. And if you culture the cells, and the process of introducing these 4 factors is actually called reprogramming. So after reprogramming and culturing the cells for 20 days, what you'd notice is formation of embryonic stem cell-like colonies. Now these colonies in this work were picked up and expanded, which means that if they could be expanded, they had the characteristic of replication. Then there was characterization and really validation. Are they really stem cell-like cells? Remember, we are starting from fibroblasts, skin cells. So interestingly, or as Yamanaka, alongside Yamanaka's work, we were able to replicate what he had done using different iPS cells. And to test whether or not these cells were actually pluripotent, if the iPS cells are injected into nude mice, the ones that are have no immune system, what happens is we do have formation of teratomas. I don't know if you're able to see this, okay? they have formation of teratomas. And once you isolate or sacrifice and create slides of the teratomas, you will identify cells from the three germ layers, the endoderm, mesoderm, ectoderm, which goes to show that initially the cells we started with as fibroblasts have been able to now have a stem-like cell characteristic where they are differentiating into the cells of the three germ layers. Now, additionally, now this is work that is being done in vitro. What about on the dish? For purposes of a continuation of some of the work we have done using the rat iPS cells, you are able to induce formation of neurons. Here we focused on neurons. So we started with rat skin cells, but if you expose this rat fibroblasts, form the iPS cells, and then expose the iPS cells on specific conditions, you're able to generate neurons, which went to prove further the supporting factor that the iPS cells are actually pluripotent. This is simply some of the genes that are present or expected to be present in neurons that initially were absent from the PCR, we can see here, and are now expressed. And this, you can even see the dendrites and the elongations and the beautiful neurons that are formed from what was initially fibroblasts. Now, for that was the rodent, and that was mostly the initial work that was done. Now, from this point forward, many of the stem cell scientists, we started to work on the human cells. So there was the same phenomenon that we had observed in the rodents, whereby an isolation of a fibroblast from an adult or from a human, doesn't have to be an adult, sorry about that, and reprogramming with these 4 Yamanaka factors resulted into iPS cells. Now, these iPS cells, when exposed to certain specific conditions, were able to differentiate into either cardiac muscles, red blood cells, skeletal muscles, neurons, skin cells, you name it. If you have the right conditions, you can be able to come up with almost any cell in the body apart from the gametes. Okay, and that is why they are not totipotent, but they are induced pluripotent cells. So what are the applications of these pluripotent cells? The first application and what comes to mind, and was the first sort of initial reason why everyone was excited about the iPS cells, is that you could take a fibroblast from a patient, create iPS cells, and if you simply think about it, from the patient's own iPS cells, you could replace or create neurons, which would be transplanted to the patient. As I've given an example of a patient with stroke or spinal cord injuries. We're talking about hepatocytes, patients who have liver cirrhosis or liver damage. Why don't you grow them a new liver? Okay, so that is the first application, and that is under regenerative medicine. Now, leaving regenerative medicine, there are other things or usefulness of these iPS cells, and especially patient-specific iPS cells. When we have patient-specific iPS cells, it simply means we are able to, one, for example, go through gene editing, which is a new tool that we have, correct any mutations that were there, then culture the cells again, differentiate them, and transplant them back to the patient. Okay, so whereby you're correcting some genetic mutation or genetic abnormality that the patient has, that is cell therapy. Now, for us as pharmacists, what might be more interesting or relevant would be from the patient-specific iPS cells, we could develop disease-relevant cells, whereby we are studying the mechanism of diseases. We are looking at targets and validation of different drugs. We're going to see this in the next slide. And also just for the simple study of developmental biology. So these are some of the applications of the iPS cells. Now, this is an example of Alzheimer's disease or Parkinson's disease, whereby you'll have this patient's skin fibroblasts. From there, you reprogram them and produce diseased. Remember, they're from the patient, so they're diseased iPS cells. If you have a way of correcting the mutation, correct the mutation, create healthy iPS cells, differentiate those cells into healthy neurons, transplant them to the patient, and you're able to have managed or treat their patient's Alzheimer's disease or replenish the dopaminergic neurons that may have gone have degenerated in Parkinson's disease. The other side we can look at here, we have the diseased iPS cells. Let's create diseased neurons. Compare those with healthy neurons. Look at what molecular pathology might be different. And there we can come in with the development of drugs, the pharmacology of drugs, and targets different systems by comparing the diseased neurons from patients and healthy neurons. So we are trying to create a system whereby we are not using rodent models, but are able to, as much as possible, recreate the disease as it is in the patients. Lastly, this is partly of what part of what I have said till this point, but we can also have formation of organoids. Remember, if you expose your iPS cells to the right condition, we call it directed differentiation, you'll end up with specific organs. Let's say, for example, you have an organoid of hepatocytes. You're able to use that to test new target drugs, okay? And it enables fast and rapid drug screening of any lead agents that you may be having. And now, because these are disease specialized cells, it is more clear than using the animal models. But now, what is the reality? This is in the lab. Theoretically, this is what is happening or can be done and is being done. But what is happening clinically, or what is happening on the ground, for lack of a different word? I have already described this, so we are going to look a little bit more into these different levels of regenerative medicine. So when we talk about regenerative medicine and we come to the stem cells, we have the move to, we're trying to move from stem cells, from personalized medicine to universalized stem cell drugs, such that you're not creating a cure, or you're not creating, for example, neurons for a specific patient, but we are able to create off-the-shelf products that can be used by a number of people and not just being patient-specific. So we have, excuse me, we have the stem cell drugs, which are products of stem cells, and also allogenic stem cells. Allogenic, of course, now means they're manufactured in batch to batch, okay, which means quality can differ sometimes, okay. And then at the end of the day, the batches can differ in quality because of the donors differing. That is why we're trying to avoid allogenic stem cell therapy. When you talk about stem cell drugs, these are drugs that are now produced, remember they're still cells, but are produced in large quantity with consistent quality. And of course, to be able to ensure there is the consistent quality, we have the good manufacturing practices guidelines, okay, manufacturing guidelines. And this now, in this case, they will be approved as drugs because they have followed the GMP guidelines. On the other hand, allogenic stem cell therapy products normally are approved as medical devices. But now the nitty-gritty of this differs with legislation in different countries. So stem cell drugs, These are new members, of course, of the pharmaceutical products. And they're not only, what we say stem cells, and you would assume it would be only the cells. No, even components, RNA, proteins, peptides from the stem cells can be formulated such that they are also effective as the whole cell, as the whole stem cells would be. What this does is it reduces any risk of immunogenicity, of course. There is easy processing, and then there is the quality assurance, storage, and of course, easiness in delivery of the drug when you're talking about components of stem cells versus stem cells themselves. So again, we're talking about the reality on the ground, approved stem cell therapies. I'm sure we've all had this. There are many clinics everywhere that announce or put it out there, advertise that they have stem cell therapies. Well, there are quite a number of approved and or marketed regenerative medicine products under the stem cell therapy docket, okay? But what we'll notice is that they are normally country or region specific. For region specific here, especially the Japan and the America FDA have been working closely such that they have approval going simultaneously in both regions. There also are specific indication only. So we have approved stem cell therapies, but of caution here is they could be approved, yes, but for specific indications only. For more information, I like to use the Alliance of Regenerative Medicine, and this normally offers what is actually approved and for what indication the stem cell therapies are being used for or are approved for. Now, I'll give an example of the FDA. So the FDA has about 23 approved cellular and gene-based therapies. Okay. Now, at the moment, these stem cell therapies, the source of the cells are actually hematopoietic cells. They are not the iPS cells I've been talking about, and neither are they human embryonic stem cells. So there's the use of the hematopoietic cells, and these are normally isolated from the umbilical cord. I'm sure you've heard of the cord blood banks, okay, cord blood. Now, there is a list of specific indications that for which they are approved for. And now this is where the gray zone is, because patients are deceived into believing that a certain gene-based or cellular-based therapy is approved. Yes, it is approved, but not for whatever condition that that patient in particular has. So for more information, always follow the different or look at the approved products. I'll put a link here for the FDA, that is. Of course, different countries have different approved uses and approved products. Now, this is about the stem cells that we have already in the market. Now, what is happening with clinical studies? Now, there is a large number of clinical studies currently going on, and they're using, as you can see, embryonic human stem cells. I know the question will be, but you said they've been banned. Up until 2001, anything that was generated up until then can be used. So there are those colonies. We have this stands for human iPS cells. And just to go through this, some targets we can have: diabetes mellitus, there's type 1 DM. There are studies, clinical studies going on for that. Parkinson's disease. We have a lot to do with the retinal disorders, the eye, okay, to mention but a few. So there is another useful site that you could go to and look at what is going on with human pluripotent cell-based cell therapy, different clinical studies. Okay, this I put a link here and I can always share it in the chat after this. So there is quite a long list. As you can see, that even recently, as recent as 17th of October, we have a phase 1 trial of advanced hematologic malignancies treatment using cell-based therapies. Another quite interesting example, and this should be in phase 2 right now, but yes, I think should be in phase 2. iPS cell-based therapy of Parkinson's disease. For this one, we have the patient's cell fibroblasts used to create neurons, correct the neurons and transplant them back to the patient and to see if there is symptomatic improvement. This is interesting because this is the use of iPS cells in screening of different drugs, already approved drugs against RNA viruses, including the COVID virus. It was very interesting. This was recent. I believe this was published last month, if I am not wrong. We have another interesting study that showed infusion of mesenchymal stem cells in spinal cord injury patients, and there was improvement with no adverse effects, but there was neurologic improvement, okay, in 12 of the 13 of the patients. That's also another interesting study that has gone on. So there are quite a number of clinical studies that are ongoing with the use of either the iPS cells from iPS cells, or cells from the human embryonic stem cells. Remember, we are not generating them right now, but what we have as cell lines and stalks. So what is the ideal world here? I take an example from the CIRA Institute, which is an institute that Professor Yamanaka started with part of his earnings, in quotes, from the Nobel Prize he won. And this is a generation of an iPS cell stock, whereby if you're able to cover a certain number of haplotypes, HLA types, in different populations, then you'll be able to create cells, generate iPS cells, so you can stock them for distribution. And this institution is open to distributing the cells to other medical, either enterprises, research institutions, universities, and the like. And from there, we can have a generation of differentiated cells, transplantation, also just drug discovery as a whole. Now here I mentioned HLA type. HLA type, of course, is population specific. And now in Kyoto, this is in Japan, the first target they're doing, charity begins at home. So they are trying to cover their population according to their HLA types. So it would be interesting and actually important for us. Charity has to begin at home. We have to do it for ourselves to create an iPS cell stock for the Kenyan population, because what they have here cannot cover our HLA types. Great. So in a nutshell, that is my presentation today from regenerative medicine, from the lab up until to the patient, how it can be used for studies and treatment of various diseases. I'm open for any questions. Thank you for your audience.
Hello, Daktari.
I can hear you.
Thank you very much for the wonderful presentation. Just taken me, among the first slides, they took me back to my first year class.
Exactly, embryology.
Yes, embryology. That was wonderful. That was wonderful. That's a very insightful presentation, Daktari, you've made. So probably we jump in, probably allow you to grab a glass of water as we go to the Q&A session. Yeah.
Okay.
Okay.
Should I stop sharing my screen?
It's okay. You can place it there. Probably a question may arise from here. You may need to expound. Yeah.
Okay, got you.
This is your last slide. Yeah.
Yes, it is.
Okay, perfectly well in order. So, Dr. We go directly to the Q&A section.
Yeah.
And our members kindly place your questions on the Q&A tab. It is easy to follow up. First question is from Peter Owino. What is your comment on the use of stem cell therapy in the treatment of autism and ADHD? Is it effective? And is the success rate significant? Over to you, Dr.
Okay, when it comes to the treatment of autism and ADHD, to the best of my knowledge, I believe for now there is no effective treatment by the use of stem cells. However, there is research going on to identify, and the genes that are related to autism and ADHD have been identified, some of them. If those could be corrected, then you could have a gene correction even at the embryonic level. So we wouldn't be talking about effectivity here, or effectiveness, sorry, and success rate, because this is still in the lab, preclinical and in very early stages. But if you're to think about it theoretically, it is possible, especially so if you are able to combine this with the gene editing tools, such as CRISPR, where you're able to cut and join genes as you please. And Peter, just to add on here, something that I should have mentioned, you can actually, with the kind of technology we have, create a superhuman. You can sit down and say, I want somebody, if you know the gene that codes for height, the gene that codes for long hair, the gene that codes for intelligence, you can have those genes in high repeat. You don't want somebody who, you could come up with a superhuman if we were left to be able to play around with human embryonic stem cells as we'd like. So with autism and ADHD, I know there's still studies, especially to identify the genes related to those. I hope that answers your question, Peter.
Dr. That answers the question perfectly well. So we move now to Odhiambo David. This is a young enthusiastic pharmacist. He says, was the same concept applied in development of Cambria used in the treatment of cancer of the lymph nodes? That's probably lymphoma. Patient cells are obtained, multiplied, and injected into the patient with lytic properties to kill cancer cells. Was it the same for that case, Dr.?
Oh, well, I am not very conversant with this study. I would also like to mention that I am biased with the brain to some extent. So this would actually be a point for me to also look into, but there is a lot of targeting, cancer targeting treatment with the use of iPS cells. So there is a possibility that could be, it is a possibility if you think about it theoretically.
Wonderful, wonderful, Dr. So we move now to where you say that is your main area of interest, that is the brain. Dr. Soreja Goral is asking, will this help in Alzheimer's disease?
Absolutely, absolutely. And Alzheimer's disease and Parkinson's disease, neurodegenerative disorders were among the first targets. And when I talk about Yamanaka's team, Alzheimer's disease is one of the main areas they are studying, whereby we can replace these neurons that are dying from the accumulation of the Lewy bodies. So yes, this would be helpful and potentially might be one of the early breakthroughs that we'll see in stem cell drugs or stem cell therapies using iPS cell-derived cells. So Alzheimer's disease is a big hit.
Perfect, Dr. That's well in. So we move on to the question and answer. We know at Dr. Michael Mungoma, that is the chair for the education committee. He's asking, do we have facilities in Kenya growing or propagating these cells for clinical use or research?
Okay, sadly to say at the moment, the best of my knowledge, no. This is one of the things I hope to do, actually create a facility that will create a Kenyan specific iPS cell line. And if anyone knows anyone out there who is doing or interested in such kind of research, this is where we network, come up with something that, we can deliver made by our people for our people. So to the best of my knowledge, we don't have any. Also, I'd like to mention, given the fact that these are cells that are potentially going to be products that are going, of course, to be used as pharmaceutical products, the restrictions and the GMP guidelines are quite stringent. So I do understand to even set up a facility to be able to produce cells for clinical use, that will be a challenge, but financially, especially the costs. But when it comes to research, that is a little bit different. It is still manageable to some extent if we get the right players on board. I hope that answers your question, respected senior.
Perfectly, Dr. So as we move on, let me also mix in some comments. This is one from Elvis Ochieng. He says a wonderful presentation. Dr. Kwanzaa Ndemo, who is going to be soon joining the Nairobi branch, says that this will help with challenges being faced in organ transplantation. Patients who cannot find an organ can be saved.
Absolutely.
Yes.
Sorry to chime in on that one. There is quite great progress, especially with the liver. We know the liver is regenerative to some extent, isn't it? Yeah, so absolutely. There is work that has been done now. This, the last time I checked, with mice, rodents that have half liver cirrhosis, and they are given what we call organoids that have been derived from iPS cells. And within a span of 3 months, they're able to almost fully regenerate a fully functioning liver. So totally, this will help us when it comes to that.
Perfectly. Dr. David Odiambo, one of the young leaders at the Young Pharmacists Forum, says this is an opportunity for collaboration. We need to get in touch and explore options. So Dr. probably on the chat box, you can share the email address that you're free for the members to be able to get in touch with you. Also, absolutely. Dr. Shah, this is to inform you that after all our webinars are recorded and are posted on our YouTube page, that is Pharmaceutical Society of Kenya on YouTube. So we go ahead, Dr. Now to the questions. From Dr. Ashford Gideon. He says, very informative and interesting presentation, Doc. Can you kindly contextualize stem cell innovation in tumor/cancer management? Thanks. Sorry, Dr. This tumor seems more more inclined to tumor and cancer. You may be much interested in the brain, but of course you can tackle some of these questions, Dr.
No, and I get where this is stemming from. I should have mentioned the 4 Yamanaka factors. Out of those 4 factors, actually 3 of them are oncogenes. So you cannot have stem cells without, or stem cells and ignore tumors or cancers. So what is normally there, what I have seen some studies being carried out when it comes to cancer management is targeting cells that will, iPS cells come up with immunological cells or immune cells that target specific cancers. That is work that is being done. Now, on the other side, I know I did not mention this, as a downside of the iPS cells or even the human embryonic stem cells, the problems of potential of tumors forming is the biggest risk, actually. And I think it would have come up as a question, what is the risk here? Yes, the risk is development of tumors because of the involvement of oncogenes. So yes, there's a lot of targeting being studied, whereby we have iPS cells being differentiated into specific immune cells to target cancers. Yes, I hope that answers your question, Ashford, sorry.
Perfectly well, Dr. So yes. We move now to one of your colleagues. She's in for the lesson today. This is Dr. Hilda Derito. She says, explain more on allogeneic stem cells versus stem cell drugs.
Okay, so when we talk about allogenic stem cells, I mentioned that they are manufactured in large batches and they are manufactured from unrelated donors. And they are mainly, or they are stem cells. When we talk about stem cells drugs, we go a little bit further. Yes, we do have the stem cells being manufactured from various donors. However, these ones are more standardized, the stem cells drugs, because we are hoping to use them, or we intend to use them as pharmaceutical products. So they're standardized, and the GMP guidelines are higher. Apart from that, you don't necessarily have to use the stem cell when you talk about stem cell drugs. It could be mRNA from the stem cell. It could be a protein from the stem cell. So that is the difference between allogenic stem cells. These are literally the stem cells, but stem cell drugs could be derivatives from the stem cells. I hope that makes sense. That is clear, Hilda. Oh, sorry, Daktari.
Perfectly well, Dr. So we go to Dr. Peter Oweno. He's asking, related to the first question on stem cell therapy, do you know any data to support this therapy in treatment of leukemia and other cancers? To my knowledge, the success rate is just too low, but also literally of reach. How would you respond to that, Dr.
Well, the success rate, of course, is low because also there's a lot still in the one, they have a problem recruiting the patients. And yes, the success rate is low. Also remember, when it comes to leukemia, sometimes you don't know that the progress of the disease is faster than we can support the therapy with the stem cells. So this is still a bone of contention, but yes, the success rate is low. But when it comes to patients, sometimes this is the only way out. And there are an improvement, especially from the cord blood derived cells that have targeted leukemia with higher success rate than what we have normally had.
Asante, Dr. Thank you very much for your well in response to that. So now we move to the other question by Brian Waliula. Are there any specific reported adverse reactions from the use of stem cell therapy? That's probably from a pharmacovigilance specialist.
Yes, there is quite, as I mentioned, I think a couple of minutes ago, there are adverse reactions, and the highest risk is formation of tumors. And these tumors normally form because of course, I mentioned the Yamanaka factors. The transcription factors are actually oncogenes. So in some cases, these oncogenes do not turn off, and they end up forming tumors. So the main worry of adverse reaction is the formation of tumors. I know there is one study, clinical study, that had to be terminated or stopped because of formation of tumors. Yes, so that is the highest risk that we have at this point. And of course, if they are not autonomous or they're allogenic, derived from unrelated donors, there's also the risk of rejection.
Perfectly well, Dr. Thank you very much for your wonderful responses. As let's probably move forward, Dr. We're having quite a number of questions. Let's just hope you're able to swallow some glass of water, just kidogo too, so that we're able to go fast. Now we move on to a senior member of the profession. She's Dr. Alice Gishane. She says that there are instances where immune system mount attack on the stem cell-based therapies, perhaps a recognition of the reverse nature of the process. Is there any instance like such that has happened?
Absolutely.
Oh.
Absolutely, that is a possibility. And this normally happens, especially so when you're dealing with the unrelated donor. If it is from the same, stem cells have been made from the same donor, this hardly ever happens. But for different donors, yes, it is a high risk, and it does happen. There are instances of that.
Okay, perfectly well. Then there's from Aman Ashani. Thank you very much for your presentation, Dr. Juliet. In your opinion, what do you think we can do as pharmacists to help reduce the stigma surrounding stem cells and stem cell research, which I think is the biggest issue facing the development of this branch of pharmacy? Dr. One of your foot soldiers what support do you need?
Yeah. Yes, I believe one is having the correct information, putting the correct information out there. One, stem cells are not a miracle drug to start with. And also, if you have the correct information, you're able to direct your patients or guide whoever it is to the right, clinics or trials, because there are so many people out there purporting to be doing stem cell therapy, yet they aren't doing anything that is approved. And as pharmacists, I think what we can do is work also on drug development using some of these stem cell derived cells. Like if you were to come up with a drug for, let's say, schizophrenia, why don't you use neurons instead of isolating them from a mouse? Why don't you use neurons that you've come up with from iPS cells, for example? I think that will help reduce the stigma surrounding stem cells. But knowledge is key here. Knowledge is power, I believe.
Perfectly well, Dr. Thank you very much for that response. Now we are having a question from Dr. Osman Mohammed. He's asking, how does someone know the difference between stem cell fraud and actual scientific therapy?
Okay, now this will take me back, and as you've said, Dr. Ali, this is recorded. I have, I think, given 2 or 3 credited or credible, sources of information on stem cells and therapies that are actually approved. So always cross-referencing and checking information from credible sources. The FDA is one, I've given another one. I think that is the way you can tell the difference. That is the only way you can tell the difference.
Okay, perfect. Thank you very much for that.
Yeah, so just keeping in touch with the regulatory bodies, yes.
Okay, now we move ahead. Probably I wish we had somebody from PPB to probably give us an insight on are they probably listening what is happening out there now to also try and inculcate it. Yeah? So I'll look through the attendees to see if there's one person who will be able to comment. Now, from the chair of the education committee, do we have stem cell development work done in Africa and in some of our poor understood conditions like cleft lips in babies, sometimes genetic influenced personalized medicine? Dr. do you have a response for that?
Yes, absolutely. Now, at the moment, I do know South Africa is doing some work on stem cells, but as he has mentioned, genetics is key when it comes to stem cells. It just has to be for your people, by your people. Kind of situation. So as far as I know, for Kenyans, we have not started. And it is even more complicated for the Kenyan society because of our different ethnicities, the Bantus, the Kushites. That already means the genetic makeup is different. So South Africa is doing some work, but for them, apart from that, I believe we are lagging behind.
That's probably saying that in Kenya we need to probably boost our response towards that. Now, Dr. Enoch Ikonya, a young community pharmacist, is asking, can this be used in cerebral palsy in the young kids?
Cerebral palsy, yes, but it depends also. Now, I can't say yes or no, but if you look at it theoretically, and you're able to replace the damaged neurons in whatever part they are, then potentially it could be useful. But I am yet to see a study that is targeting cerebral palsy.
Thank you very much, Dr. Dr. Millicent Mo, who asks, please comment on the progress made on sickle cell management in regard to sickle cell, in regard to stem cell therapy.
Whoa. Again, there I will beg to refer my colleague. I cannot give a definitive answer on what is happening with sickle cell management. Again, I believe I am biased to the brain. So that will require reading into literature that is available.
Okay, perfect, Dr. So we now move towards the end. And what Dr. Kwansandemo, once again, what is being done to protect one's genetic information? Could you make a quick comment on the bioethics behind this science?
Great. Yes, this is a very contentious area and important, and there are very many gray zones. So when it comes to one's genetic information, to start with, there has to be the informed consent of the donor. And this is informed and signed documented consent. Also, there is the aspect of the institution ethics and research committee approving such studies, okay, of the institution. Like in my institution, we have an ERC board that will approve that. So in that sense of protecting one's genetic information, that would be my comment. And on the sense of bioethics, when you talk about stem cells and bioethics, this will never be, we will never conclude on this. There's a lot of the social aspect of it, the ethical aspect of it. It's quite a gray zone. So I believe it depends on your the institution's ethics review committee to see if indeed a study has conformed to, I mean, consent, obtaining consent in the right way. Is it within the ethical practices of using human subject information? I hope that answers Dr. Ndemo's question.
Yes, now we're having Professor Keegan. Professor Keegan wants to jump into the discourse and contribute something on sickle cell. Professor Keegan, I have allowed you to talk. Are you there with us, Professor Keegan?
I hope you can hear me.
Loud and clear, Professor.
Yeah, now, thank you very much, Dr. I have listened to a wonderful response, and I think I can help you answer some of the questions about the tumors and sickle cell. Now, what's going on? There's something Dr. mentioned about personalized medicine, whereby we can now correct defective genes. She talked about cutting them and repairing by use of such methods like CRISPR. There is now a very new one by Harvard University. It's called RRL. It's about 3 months ago. Now, what happens here is, you know, like you have adenine, cytosine, guanine. So if you have a faulty SNP, at the moment, we can only correct single nucleotide polymorphisms. And one of those conditions is sickle cell and Huntington's disease. So they have now been correctly corrected. It started 3 years ago on a 14-year-old boy, and also some retinoblastomas that are now being corrected through CRISPR technology and RRL. Now, about tumors, using this system, I know you can use stem cell for leukemias, etc. But using gene editing, it's still a problem because you have to edit a whole, section like up to 3. But people are hoping that there will be new technologies where you can rearrange and take out the mutation. We have now treated sickle cell, Huntington's disease, and several other conditions with SNPs, single nucleotide polymorphisms. Thank you. I hope I've answered some questions.
Thank you very much, Professor Keegan, for...
Thank you, Professor.
Asante, well in. I can see probably pharmacists are taking keen on the day-to-day happenings outside in the scientific field. That is quite encouraging. So we probably now conclude on the Q&A. And on the last question was by Samson Aluda, Dr. Samson Aluda. He says, do we have practical scalability stroke application of human stem cells, as well as the challenges, if any, of using automation and bioreactors to produce the quantities, stroke quality of cells required for clinical and biomedical use? Dr.
Yes, absolutely. And I think towards the end, I did mention one such facility in Kyoto University, the CIRA Institute, whereby they have actually managed to do this. Of course, it is very costly, and given the level of the GMP conditions required, it is costly to ensure the quality. But yes, I believe the CIRA Center has been able to create such a system. So it is practical and applicable, and it can be done.
Wonderful, wonderful. Probably the last question from Dr. Mureithi Mungiria. He asks, can it be used to reverse aging, wrinkles, metabolism? Also, can it be a solution to infertility?
To reverse editing, the reverse aging, wrinkles, metabolism. Well, this all stems down to what Professor also mentioned, and that is gene editing, whereby you can cut and fix and like tailor make your genes. If that was possible, then theoretically, you could, I don't know, aging has to do with the telomeres. So I don't know about reversing aging, but you could do quite a number of things with this technology. Solution to infertility. Well, now infertility is of different causes. So I find it difficult to comment on this one specifically. But what I can say about especially the iPS cells, we have still not been able to create gametocytes from iPS cells. So in that sense, in relation to fertility, that could be my comment, but I believe infertility is of various causes, so I cannot give a straight answer to that.
It's okay, Dr. Thank you very much for that. You've taken your time to probably ensure that we are well brought up to par in matters stem cell. From my end, I was probably a true novice in these regenerative medicine course, and probably even for some of the community pharmacists who are also the ones who used to the community setup are not able to, were at least able to appreciate some of the difficult conditions they are treating at the community pharmacy level and also at the hospital, the clinical pharmacist. But there is hope. But now, Dr. for you, now the main thing lies, when are you willing to introduce this in the country?
I've already identified a collaborator in Prof.
Okay.
Yes. Now it is for us to set up teams and come up strong. And I mean, here the funding is of importance, of course, because this is not a cheap venture, as is very many of the pharmaceutical ventures that are there. But collaborations, I believe, first is key, and awareness. I believe some of the students I've taught, they had never heard of such, and their interpretation of stem cells was totally different. So also awareness and the like. But I see a different horizon for me. The next 5 years should be different. We'll be having a different discussion from this day. At least we should be talking about Kenyan-derived iPS cells.
Perfect, well, Dr. I can see some of our seniors, Dr. George Matimbai, the chair for PSK Western Branch, has listened in. Probably he will use his acumen in convincing patients and also recruiting for you patients. Also, in matters of pharmacovigilance, I can see you're sorted because Dr. Jayesh has been with us in these meetings. So we're having people who have listened and are keen to ensure that once you hit ground and you're out there, Dr. and are interested in looking for the right people to drive it. You can see even Professor Keegan is there. Professor Gabriel Keegan is there to even support you on that.
Absolutely, thank you.
Yes, so probably I have 3 people who have raised their hands. There's Dr. Stephen Twitter and Dr. Joan Wanyoto. Probably I allow you to talk if you have a question as we wind up. Dr. Joan Wanyoto, Dr. Emmanuel Mokua, and Dr. Stephen Twitter. Any questions from your end? Oh, perfect, Daktari. From my end, Daktari, from the Pharmaceutical Society of Kenya, of which you are a member, Daktari, from the president of the society, Dr. Louis Mashuku. Emmanuel Mokua, you have a question?
I'm okay, thank you.
Perfect, Emmanuel. Dr. Joan Wanyutu?
No.
Okay, Dr. Stephen Twitter? Perfect. Let me allow Professor Keegan to probably give his parting shots. Professor Keegan?
Yeah, thank you very much. I think this was a very good talk. I just want to comment on something. I teach at a medical school. I teach medical students. We don't have a pharmacy school in Eldoret. But now there are other issues that Dr. has mentioned that we're already teaching our medical students. I hope they can be taught in the pharmacy school as well. Like, personalized medicine, the way we used to talk of slow and fast acetylators, etc. Now they're in practice. And very soon we shall be training pharmacists to have personalized medicine so that you don't have to ask a patient, ukimeza hii utajikuna, and exactly. And it can be part of the differential. So I think this is very entertaining and we should have more of this. Thank you.
Asante sana, Prof, for taking your time to guide us through that. So as from the Pharmaceutical Society of Kenya, from the President, Dr. Louis Mashogu, receive his regards from the National Governing Council, the National Executive Council, also received the regards of the CEO from PSK. We are much humbled for you for continuously joining our meetings And also, this is to alert our members that if probably you are in a field that you feel that our members are not up to par, kindly reach out to me. You have my number, you have my email address. Reach out to me. Let me know how best we can organize to ensure that we have such kind of forums to enlighten each other. We are totally open to ensure that anybody who has knowledge, not compulsorily be scientific, even if it's on the business side, you have knowledge we would like to share with us. As long as it's not commercial, we will allow it from our members. It's absolutely free of charge for our members. So ladies and gentlemen, without further much ado, let me probably announce to you that we are having the conference in Mombasa. Probably Dr. Juliet Makanga, it's my prayer and hope with Professor Keegan that you combine and probably have one presentation for us during the conference at Mombasa.
Yeah? Okay, absolutely.
Yes. The conference is going to be taking place on the 26th of October to the 29th of October. The conference is going to take place from the 28th to from the 26th to the 29th of October. So let's be prayerful and also take care of ourselves as the fourth is taking a toll on the nation. So let's be very cautious so that we're able to get together and meet at the conference. So ladies and gentlemen, I wanted to give you the theme for the conference this year. I don't want to leave you without perfect. So the theme for the conference this year is advancing the health and well-being of Mwananchi through excellence in pharmacy. Dr. Juliet Makanga, are you set for that?
I'm sure there's a place we can plug in.
Yes, Professor Keegan, you see there's a place that you can plug in, advancing the health and well-being of Mwananchi through the excellence in pharmacy. The call for abstracts?
Yeah, so maybe we can, sorry, my apology. We have also started a master's program in clinical pharmacy in Eldoret. So I'll check maybe if one of the students will be traveling to Mombasa, but we can do a quick paper on pharmacogenomics and personalized medicine. If I can get the contact or email of Daktari, and then we can do one paper, both, because they are all related.
Perfectly well, Daktari. Oh, wonderful. So ladies and gentlemen, without further much ado, I also want to announce that the call dates for the abstracts are going to come out soon to all of you. So just be ready and prepared. Na namba jinsi nisemavyo kwamba lililo na mwanzo lazima aliwe na mwisho, mpanda ngazi hushuka. Nikubali ni warudishe kidogo katika niwakumbushe kidogo katika zile insha mlikuwa mnaandika, mnasema kwamba waganga wakutupaka mafuta kwa mgongo wa chupa waliponena lilo na mwanzo lazima liwe na mwisho. Tumefika tamati ya somo letu la leo. Nitawauliza kwamba muhakikishe mmeweza kubonyeza katika portal ya Pharmacy and Poisons Board and subscribe to today's meeting. Otherwise, we meet next week on the 3rd of August. We are always on Tuesdays and Thursdays. Sina mengi ila kuaga na kuambia alamsiki. Have a great evening and God bless you.
God bless you too, Dr.